So in this video we are going to see about the structure and function of skin. So the skin is the largest organ with surface area of about 2 meters square, and it comprises about 16 to 20% of the total body weight. So this is the structure of skin. It comprises of three components: epidermis, dermis and the subcutis, and the subcutis is otherwise called as hypodermis. So this epidermis, it consists of 5 layers: stratum corneum, granulosum, spinosum, basal, and another layer called stratum lucidum present in acral areas like palms and soles. So beneath the epidermis we have the dermis, and then beneath the dermis we have the subcutaneous tissue. So this dermis is of two types. One is papillary dermis and another one is reticular dermis. So both are separated by the subpapillary plexus. So this epidermis, it consists of cells like keratinocytes, Langerhans cells, melanocytes, Merkel cells, and some unmyelinated axons. So of all these keratinocytes, they belong to more than 90% of cells in the epidermis. And this dermis contains endothelial cells, neural cells, fibroblasts, dendritic cells, mast cells, etc. So the stratum basal is a layer of cuboidal cells, and above that you have the stratum spinosum, which is a layer of polyhedral cells. The stratum spinosum, it has many spine-like desmosomal connections visible in histopathological examination, and hence the name spinous layer. It is otherwise called as prickle cell layer. So above the stratum spinosum, we have the stratum granulosum. It is of irregular size and shape. We have this keratohyalin granules, that is lamellar granules in it. And above the stratum granulosum, in the acral areas alone, you have this stratum lucidum, which is a grayish blue structure. And above the stratum lucidum, we have this stratum corneum, which is an anucleated structure. So in the stratum basal, we have Merkel cells, melanocytes. In the stratum spinosum, we have this Langerhans cells, and here they are the lamellar granules in stratum granulosum. And this stratum corneum, it contains dead keratinocytes, that is anucleated cells. So this adnexae is nothing but part surrounding an organ. It extends from epidermis into dermis, and it consists of specialized cells for hair growth, stem cells, and temperature regulation. Next is the subcutis. The subcutis is a layer present beneath the dermis, and it forms an underlying cushion, and the cells are engorged with lipid and nourished by vessels. So the human skin, you can divide it into two types: hairy and non-hairy skin. This non-hairy skin is otherwise called as glabrous skin, and it consists of a series of ridges and grooves, which is called as dermatoglyphics. So the origin and development of skin. So this origin and development of skin, you can divide it into three stages: specification, morphogenesis, and differentiation. So the specification, it lasts up to 2 months. So the ectoderm, it gives rise to the epidermis, and the mesenchyme, it gives rise to the dermis. In this, many cell-to-cell interactions and protein signals play an important role, which help in the future patterning of skin. So this is how it will be like. This is the ectoderm, which give rise to epidermis, and below that you will have this mesenchyme, which give rise to dermis. So this morphogenesis, it takes about 2 to 5 months. It forms specialized structures. In this, the stratification of epidermis and the appendages, vascular formation, everything takes place. So the differentiation, it takes from 5 to 9 months. There will be further development to form a mature skin. So initially, during the 3rd week of intrauterine life, the development of skin takes place. As I told already, the ectoderm, it gives rise to the epidermis, and the mesenchyme, it gives rise to the dermis. So at the 3rd week, initially there will be only a single layer of glycogen filled cells, and what happens is around the 6th week there will be 2 layers. Below you have the mesenchyme, and here you have the ectoderm, and the second layer will form, that is called as periderm. So next, around 8 to 11 weeks, what happens is, in between the ectoderm and periderm, another layer develops. So this is the periderm and this is the ectoderm. In between the ectoderm and periderm, a middle layer of cell develops. This is from 8 to 11 weeks. So next is around 12 to 16 weeks, the middle layer of cells, they increase in number and size. That is between the periderm and the ectoderm. This is the middle layer of cells. This is the periderm and this is the ectoderm. There will be many middle layers and microvilli, and these middle layer of cells, they contain mitochondria, Golgi complex and tonofilaments. So this entire process, it requires the presence of P63. P63, it is an epithelial immunohistochemistry marker, and the mutation of P63 can lead to conditions like ectodermal dysplasia. Yes. So around the 10th week, you can see the desmosomes and hemidesmosomes, and around 14 weeks there will be basal keratins, and by 15 weeks there will be formation of filaggrin. So this filaggrin is nothing but filament aggregating protein. So next, the keratinization of the appendage, it is completed by around 11 to 15 weeks, whereas of the epidermis, the keratinization is complete by around 20 to 20 weeks. So around 21 weeks, we can see the keratohyalin granules, and by 24 weeks, so the periderm, which is present above the middle layer and the ectoderm, what happens is, at birth, this periderm, it starts to separate from the, around 24 weeks, the periderm, it starts to separate from the embryo. So at birth, along with the shed lanugo hairs and sebum, this periderm also joins and forms the vernix caseosa. So coming to keratinocytes. So how keratinocytes differ from other cells is by their large size and ample amount of cytoplasm and the presence of intercellular bridges. So it has 4 layers: corneum, granulosum, spinosum and basal, and the 5th layer, stratum lucidum, is present in palms and soles. So this is the normal basket weave architecture present in non-acral sites. You can see it is anucleated, and this is the thick and compact stratum corneum present in areas like palms and soles, where also you can see the grayish blue structure, that is the stratum lucidum. So next, coming to the stratum basal, this is a layer of cuboidal cells about 1 cell thick. It contains more basophilic cytoplasm, so it is attached to each other by desmosomes and to the basement membrane zone with the help of hemidesmosomes. So this layer contains keratin intermediate filaments, which along with the cytoskeletal proteins helps in the formation of stratum corneum. So another unique feature of this stratum basal is the mitotic activity. This layer is capable of continuous differentiation. So in order to see the mitotic activity of stratum basal, you either need the thymidine labeling of cells in the S phase of DNA cycle or Ki67 in the paraffin sections. So the cell divisions, it occurs every 18th to 19th day. There are 3 types of cells in stratum basal. So one is stem cell, number 2 is transient amplifying cells, and number 3 is post-mitotic cells. So next, coming to stratum spinosum, as I told already, they are polyhedral cells around 5 to 10 layers thick, whereas the basal layer is only 1 cell thick, that is 5 to 10 layers thick, and it is also called as prickle cell layer. So if you see, there are many spine-like margins in histopathological examination because of the abundant desmosomal connections, hence the name stratum spinosum. So in the intercellular spaces, there are many glycosaminoglycans, of which this hyaluronic acid is the predominant one. So it contains many granules called as lamellar bodies and intercellular cement substance called glycocalyx. So these tonofilaments, they are nothing but the keratin intermediate filaments. Their one end is attached to the desmosome, whereas the other end is present within the cytoplasm. For example, this is the cell inside, and this is the desmosome, that is the intercellular bridge connection. Inside you have the keratin intermediate filaments. Their one end is connected to this desmosomes, whereas other end is connected to the cytoskeleton within the cell. So this desmosome on electron microscopy, it is visible as two dense desmosomal block and a multi-layered middle zone nanometer. Here this is the architectural structure of desmosome. So this desmosome also contains calcium dependent cell adhesion molecules called as cadherins, which are these desmogleins and desmocolin. So here you can see this is the desmoglein and this is the desmocolin. So this desmoglein and desmocolin, they are linked to the intracytoplasmic filaments with the help of placoglobulin, placofilin and desmoplakin. So autoantibodies against any of these structures can result in pemphigus. So this desmoglein 3 is mainly present in the immediate suprabasal keratinocytes, whereas desmoglein 1, it is preferentially distributed beneath the stratum corneum. So next, coming to the stratum granulosum, it is otherwise called granular layer because it contains intercellular basophilic keratohyalin granules. They are of irregular size and shape and about 2 to 5 layers thick, but it is variable depending upon the location because in certain areas like palms and soles there can be up to 10 layers thick. So during keratinization, the keratohyalin granules, they contain two structures. One is the interfibrillary matrix or filagrin and another one is the marginal band. This filagrin is nothing but filament aggregating protein which helps to keep the keratin filaments together, and this marginal band, it forms a lining around the horny layer. So this tonofibrils, they are soft and flexible. The tonofibrils, they are nothing but aggregation of tonofilaments, that is keratin intermediate filaments. So in the epidermis, it contains soft keratin, whereas skin, hair and nails, they contain hard keratin. So the soft keratin, the desquamation occurs by enzymatic action regularly, whereas in hard keratin, that is in hair and nails, they lack the keratohyalin granules, hence there will be no desquamation regularly and they require periodic cutting. So this granular layer, there will be mature keratin forming the transitional zone. Why it is called as transitional zone? Because in this layer there will be dissolution of nucleus and other organelles before forming the stratum corneum. So how this dissolution of nucleus in other organelles takes place is with the help of lysosomal enzymes, this acid phosphatase and aryl sulfatase, because they are responsible for the autolytic changes in the granular layer. So this granular layer contains lamellated granules called as Odland bodies or membrane coated granules. So these lamellated granules, they are also seen in the uppermost spinous. So next is the stratum corneum. It is the outermost layer of skin around 20 to 25 layers thick, and they are the largest cells in the epidermis. They are anucleated cells, and in formalin fixed specimens, what happens is there will be shrinkage of the cytoplasm and they form a shell along the membrane, and this leads to the characteristic basket weave architecture of the stratum corneum in formalin fixed specimens, whereas in fluorescent stain they are arranged in orderly vertical stacks. So the lipids released from the lamellar bodies, they surround the cells in the stratum corneum and they give an important role in permeability of this layer. So the stratum lucidum, it is an electron lucent layer. They are also still nucleated and called transitional. So this is the epidermal differentiation. This is the stratum basal and this stratum spinosum. So this upper stratum spinosum and stratum granulosum, they contain these lamellar granules. So coming to epidermal proliferation and differentiation, it depends upon multiple extrinsic and intrinsic factors. So normally the keratinocyte maturation from basal to horny layer is about 30 days, but it is altered in certain diseases, for example in psoriasis. So the transition, how it happens is, first from ovine basal cells, they become polyhedral cells in the spinous layer, and then they become flattened cells with granules in the stratum granulosum, and then fully keratinized cells without nuclei in the stratum corneum. So from granular layer to stratum corneum, the process is very abrupt. So the process normally requires three structures: tonofilaments or keratin intermediate filaments, keratohyalin granules, and the Odland bodies. So even in epidermis like mucosa, The stratum basale plays a unique feature in maintaining the cell division. That is, there is a continuous cell division. So this basal layer, it produces extracellular matrix, which is an important part of the basement membrane zone. So this basement membrane zone, it separates the epidermis from the underlying dermis. So the important layer of this membrane, the extracellular matrix, is formed by the basal layer. So this extracellular matrix, the most prominent protein is laminin 5, for which you need alpha 3 beta 1 integrin for its assembly. So the keratinocytes, as mentioned already, they are ectoderm derived cells and they contain keratin filaments or tonofilaments and cytokeratins. They can be acidic or basic, though acidic is type 2 and basic is type 1. So the tonofilaments, they are normally 8 nanometer thick, and the keratohyalin granules, they are usually composed of profilagrin, loricrin and keratin filaments. So the filagrin is a histidine rich protein. So the involucrin, it is a structural component of the squamous epithelium and it is present in the marginal band. So this marginal band forms the inner lining of the stratum corneum. So Odland bodies, otherwise called as membrane coating granules, lamella granules or keratinosomes. So these Odland bodies, they are discharged from the granular cells into the intercellular space and they have two main functions. One is they form a barrier to water loss. Another one is they mediate the cell-to-cell cohesion in the stratum corneum. So the regional variation of epidermis. So in places like mucous membrane of mouth, they possess neither stratum granulosum whereas nor the stratum corneum. Exceptions include the dorsum of tongue and the hard palate. So they contain more amount of glycogen, less amount of desmosomes, and they are held together by amorphous intercellular cement substance. Whereas in acral skin, you have the thick stratum granulosum and stratum corneum along with stratum lucidum. So the specialized structure and function, you have four main functions. First one is cellular cohesion. So this keratohyalin granules, what happens is, these are the keratohyalin granules, and inside the keratohyalin granules, you have this profilagrin. So the keratohyalin granules, they increase in size. These are the tonofilaments and they surround the tonofilaments. Now what these tonofilaments do is, a group of tonofilaments, they aggregate to form the tonofibrils. Okay, that is the mature keratin. So to keep the tonofilaments together, you need filagrin in between them. And this filagrin, it comes from the profilagrin present in keratohyalin granules. So what happens is, this is the keratohyalin granules and inside we have this profilagrin. By dephosphorylation, this profilagrin comes out and from that profilagrin you get filagrin. So this filagrin, it helps in the aggregation of, these are the tonofilaments, and the filagrin, it helps in the aggregation of these tonofilaments. So filagrin mutation plays an important role in atopic dermatitis. Next is the barrier function. So first one is cell to cell cohesion. Second one is barrier function. As mentioned already, this Odland granules, they fill up the intercellular spaces and help prevent water loss. So they contain mainly sugars, hydrolytic enzymes and free sterols. So in stratum granulosum, if you see, there are free sterols and sugars, whereas in stratum corneum, there are no sugars, but they contain free sterols along with neutral lipids. All are linked to each other. Another one, this lipid is also hydrophobic, which plays an important role in the barrier function. And they have some natural enzymes like steroid sulfatase, which remove lipids from the stratum corneum and they help in desquamation regularly. So next, third one is the enzyme activity. So already mentioned is these lysosomes, they contain hydrolytic enzymes like aryl sulfatase, acid phosphatase and beta lactosidase. So these lysosomal enzymes, they are more in granular and horny layer. That is, the stratum corneum is otherwise called horny layer. But also they are seen in basal, spinous and lamellar granules. Another one is this secondary lysosomes, they are otherwise called as phagolysosomes. They are present in lower epidermis and they digest the phagocytosed melanocytes. And the immune function of keratinocytes is they produce interleukins like 1 alpha, 1 beta, and then colony stimulating factor, interferons, tumor necrosis factor, transforming growth factor, and other growth factors like platelet derived and fibroblast growth factor and melanocyte stimulating hormone. So, immunohistochemistry, there are 54 functional keratin genes, and it's of two types, type 1 and type 2. Each contains 27 genes. So, keratin 5 and 14, they are mainly present in basal cells, and the mutation in keratin 5 and 14 give rise to epidermolysis bullosa. So, the basal cells that contain stem cells, they also have keratin 15, and this keratin 1 and 10, they are present in suprabasal cells, and the mutation of keratin 1 and 10, it gives rise to bullous ichthyosiform erythroderma and epidermolytic hyperkeratosis. And this keratin 2, it is present in terminally differentiated keratinocytes, and their mutation gives rise to bullous ichthyosis of Siemens. Whereas this keratin 9, it is present in the palmar and plantar epidermis, and the mutation of keratin 9 can give rise to epidermolytic palmoplantar keratoderma, that is Warner syndrome. So keratin 4 and 13 mutation can give rise to white sponge nevus, and this keratin 6A and 16 mutation is paranoia congenita type 1, whereas keratin 6B and 17 paranoia congenita type 2. So 6A is type 1, 6B is type 2. And the epithelial immunohistochemistry marker I mentioned already, it is P63. So their mutation can give rise to ectodermal dysplasia syndrome. So this P63 is overexpressed in conditions like squamous cell and basal cell carcinoma.